Good day, this is Professor Will, CBMD, PhD. Today is Thursday, March 21st, 2019. It's 22:17, 44 seconds Eastern Standard Time. And today I'm going to be doing a reading on the fundamentals of virology. So let's get started here. Virology, classification, characteristics, replication, viral diseases. Definition and properties. Filterable, obligately intercellular, not able of individual metabolic activity, energetic metabolism, and proteosynthesis. Possible only with host cell structures. Replication is not division but assembly of subparticles. Genome is or RNA or DNA. Consequences of characteristics. Viruses are not living, must be infectious to survive, must be able to use host cell mechanisms to produce cell structures or reactions, mRNA protein copies of genome, must be able to encode processes not available in host cell, subparts must be able to assemble. Classification and nomenclature. ACC to structure, shape, morphology. Picornaviridae oki, biochemical properties, RNA, DNA, disease they cause, VHA, hepatitis A, virus transmission, arbovirus, arthropod-borne, host cell, HPV, HIV, SIV, tissue tropism, adeno-entrove. Structure and properties of non-encapsulated viruses. Protein properties, stable against temperature acids, proteases, detergents drying leaves host cell by its lysis. Consequences. Spread by stool, dirty hands, dust, small droplets. After drying, they are infectious, surviving in unfavorable conditions and cold and resistant to detergent stimuli, production of antibodies, hormonal immunity. Structure and characteristics of encapsulated viruses. Membrane, lipid protein, glycoprotein properties, lability against outside conditions. Modi cell membranes during replication, leaving cells by budding from host cell. Consequences. Require human environment, do not survive in GIT, spreading by big droplets, bloodway, secretions, stimuli, cell immunity, sometimes hypersensitivity, immunopathological consequences. Primary classification. ACC to structure of virion or nucleic acid, RNA or DNA, SS or DS, segmented or non-segmented genome, linear or circular, symmetry, isohedral, helical complex, encapsulated or non-encapsulated, number of capsomers, non-conventional viruses. Replication of viruses. Stages. 1. Recognition of target cell. 2. Attachment. 3. Penetration. 4. Encoding. 5. Synthesis of early mRNA and non-structural proteins. Replication of genome. Of late mRNA and structural proteins. Assembly of parts. Budding of non-encapsulated viruses. Release from cell. Replication of viruses. Host cell is a source of substrate energy and parts important for synthesis of viral proteins and replication of genome. Struggle for energy and sources. What cell will not give as a source? That must be produced in place, must be encoded in the genome of virus. Replication of cycle, early phase of infection, recognition, attachment, penetration, encoding, release of the genome from the nucleus. Late phase replication of the genome macromolecules, assembly, release. Phase of eclipsia, from uncoding of the genome, loss of infectious until the assembly and appearance of new virions. Latent phase from eclipsia till release of virions. Recognition and attachment. Interaction of viral surface proteins, VAP, virus attachment proteins with receptors on host cell, identify target host, specificity of virus, and target tropismus. VAP encapsulated virus, glycoproteins, GP120 HIV, and non-encapsulated parts of capsid. Entry penetration interaction of VAP and receptors starts internalization, non-encapsulated endocytosis, encapsulated endocytosis or fusion. Uncoding. After internalization, genome must enter to the place of replication. DNA, EXC, poxvirus, and nucleus RNA are in the cytoplasma, endosomes, lysosomes, production of enzymes. Synthesis of macromolecules. Virus must produce an mRNA proteins and generate identical copy of own genome. Transcription, translation, and replication. Genome is useful if it is transcribed to functional mRNA that is able to bind ribosomes and translate information to proteins. This depends on the structure of genome and on the place of replication. Classification. ACC to replication strategy. DNA virus replicating in the nucleus, they use DNA-dependent RNA polymerase of the host cell. DNA viruses replicating in the cytoplasm, pox virus, produce important enzymes for transcription and replication and production of mRNA. mRNA to RNA viruses. Some viruses of resemblable structure have different ways of replication. DNA virus, transcription of DNA in nucleus. In brackets, EXCL dot pox. Viral DNA is similar to host cell DNA. DNA is labile, genome stays in an infected cell, produce commonly persistent infection. Early genes, non-structural proteins, enzymes needed for proteosynthesis, polymerase. Late genes encode structural proteins needed for assembly. Regulation, availability of DNA polymerase substrate. RNA viruses. Replication and transmission is similar. Viral genome is or is like mRNA plus RNA, or is the template for mRNA minus RNA. dsRNA is produced. Structure that does not exist normally in non-infected cells. Encoding of RNA-dependent RNA polymerase, rapidly degradable, are present in active stage after uncoating or uncodes early enzymes. Are labile, replicating in cytoplasm, easily mutate. Plus RNA acts as mRNA, binds on ribosomes and proteosynthesis, starts directly minus RNA. Dependent RNA polymerase is synthesized and enables production of minus RNA. Copy equals dsRNA. Minus RNA is non-infectious. Polymerase must get into the host cell so that the mRNA can be produced. Replication is done in the cytoplasm. EXCL, influenza virus. dsRNA retrovirus cannot produce mRNA. Cytoplasm contains RNA-dependent DNA polymerase. Synthesis of viral proteins. Viruses are dependent on host cell ribosomes, tRNA, and production of proteins. Eukaryotic ribosomes bind on mRNA and produce continual protein polyprotein that is changed by proteases to functional proteins. Post-translational modification, phosphorylation, glycylation, acylation. Assembly. Unique parts of virions assemble like three-dimensional puzzles. DNA, EXCL, pox viruses, nucleus proteins must be transported from cytoplasm to nucleus. RNA viruses and pox viruses assemble in cytoplasm. Non-encapsulated viruses, empty procapsid will be filled with genome, or capsomers are added one by the other around genome. Encapsulated viruses gain capsule during budding throughout the membrane of ER nucleus or cell. Releasing from cell after lysis from the cell, non-encapsulated viruses. Exocytosis, budding from plasmatic membrane encapsulated. Released viruses are usually responsible for new infections, sometimes for production of multinuclear giant syncytia or vertical transmission of infection. Genetics of viruses. Mutation changes the characteristics of daughter virus in comparison of wild type. Mutation of general genes. Inactivation of virus, lethal mutations. Mutation of other genes, change the properties. Deletion, attenuation of properties, changes in the host cell or target tissue, resistance to temperature. Induced chemically by radiation. In nature, they are caused by insufficiency of viral polymerase, more common in RNA and DNA. Genetics of viruses 2. Recombination, co-infection of two similar viruses, viruses with segmented genome, reassortment, assembly of defect virus with wild virus complementation. Selection pressure on new strains or mutants, possibly to survive in the host cell. Viral diseases. Transfer via natural barriers, avoidance of immunity, control killing of important cells, or production of destructive immunity or inflammation reaction. Immunity reaction is the best therapy of viral infection and also the most powerful factor of pathogenesis of viral infection. Tissue tropismus. Different diseases can be caused by the same virus. One virus can produce different diseases. Infection of target tissue. Entry to organism through skin or mucous barriers protected by tears, mucus, epithelium, stomach acid, IgA. Inhalation, most common ways of transmission of infection. Replication in infected cells where it remains or spread by bloodstream, by MFS cell, by lymphatic ways, by neurons. Viremia, primary, secondary. Pathogenesis of viral infections depends on virus on the cell. Absorptive infection, non-permissive cell, viral mutants not able to multiply. Lytical permissive cell virus able to divide. Persistent, chronicle, latent, recurrent. Transforming semi-permissive cell enable only some stages of replication. Replication leads to cytolysis or alteration of the cell. Oncogenic viruses. Some DNA viruses or retrovirus start persistent infection that enable stimulation of uncontrolled growth of cells, transformation or immortalization, continuing growth, loss of contact, intracellular inhibition of the growth ability to grow, and suspension or on semi-solid agar. Different mechanisms of inhibition to program death of the cell, apoptosis. Viral transformation is the first stage, usually is not enough for oncogenesis or tumorigenesis, but cell are more prone for oncogenesis. Immunity mechanism, nonspecific, interferon, alpha and beta gamma, antigen-specific humoral immunity, T-cell immunity, immunity reaction primary, secondary, mechanism how viruses evade immunity, immunopathological processes, hypersensitivity and inflammation. Viral diseases, acute infection, prodromal stage, clinical stage, reconvalescence, influenza, acute infection with latent complication, SSPE, late infection, VZV, chronic infection, chromal VHB, chronic infection with late onset of the disease, HTLV, show infection, unconventional viruses. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, March 21st, 2019. This is the ending of my reading of Virology and Fundamentals. Hope you enjoy listening. Have a great day.